In this video, we are going to discuss the light-dependent stage of photosynthesis. And by the end of this lesson, you should be able to state the precise site of light-dependent and light-independent stage of photosynthesis, outline the role of water in the process of photosynthesis, describe non-cyclic photophosphorylation, and finally outline the products of the light-dependent stage of photosynthesis. Okay? Before we discuss all of this, let us first look at a chloroplast because it is the site where photosynthesis takes place in a leaf. Now, when we talk of a chloroplast, we are talking about an organelle which you are already seeing on the screen, and the organelle has got an oval shape, as you can see, with two membranes. The chloroplast envelope has got two membranes, the outer membrane and the inner membrane, between which is a space called intermembranal space. Okay? Now inside the chloroplast, we have a gel-like substance called stroma. So here, this space you are seeing here is filled with a gel-like fluid called stroma. And within stroma, we have ribosomes, which are sites of protein synthesis. We also have enzymes, which catalyzes reactions of the light-independent stage of photosynthesis. We also have the starch granules that temporarily stores the manufactured starch. Okay? But in this lesson, we are not going to focus on the reactions of the light-independent stage, but instead, we are going to focus on the light-dependent stage of photosynthesis, which occur on thylakoids. These ones are called thylakoids. Thylakoid, 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 thylakoid. So these are all thylakoids. Now this stack of thylakoids is known as granum, and plural grana. Okay. Now this grana has got thylakoids which have membranes. Okay, as you can see here. So this is a thylakoid, a thylakoid, a thylakoid, a thylakoid, and they have membranes. These are membranes. And inside we have the thylakoid rumen. This is a thylakoid rumen, a thylakoid rumen, a thylakoid rumen, a thylakoid rumen. Okay? Now, the whole process of light-dependent stage of photosynthesis occur on these membranes as we are about to discuss together. So I'll be referring to this diagram in our discussions. Don't be caught offside. So in summary, the light-dependent stage of photosynthesis occur on the thylakoid membranes, while the light-independent stage of photosynthesis occur in the stroma. Together? Okay. Now, how does the light-dependent stage occur? Let us look here at the light reaction of photosynthesis, or the non-cyclic photophosphorylation. Within the membranes of the thylakoids, we have got photosystems. We have photosystem 1 and photosystem 2. Now, within these photosystems, we have got the reaction centers. Okay? Now, when a photon of light strikes the photosystem, the electrons in the reaction centers are boosted to a higher energy level. At the higher energy level, they are accepted by an electron acceptor. This electron acceptor becomes reduced when it accepts an electron. Okay? Now, these electrons that are accepted by the electron acceptor are handed over to the electron transport chain. We have proteins that are electron transporters. So the electrons are handed over to this electron transport chain. From the electron transport chain, the electrons are handed over to photosystem 1. Meanwhile, these electrons that have been removed from photosystem 2 are replaced by photolysis of water. When I say photolysis, it means breakdown of water by use of light. Photo means light. Lysis is to break down. So photolysis is the breakdown using light. Okay? So now, when water is photolyzed in the presence of an enzyme, it produces protons, oxygen, and electrons. This oxygen is given out, okay, as a byproduct. Now, these electrons here are used to replace the electrons that are lost from photosystem 2. Now, what about these protons? We are going to discuss the fate of the protons later. Now, remember that the electrons were passing along the electron transport chain from higher energy level to a lower energy level. And as they do so, they release energy. That energy released is used in the formation of ATP from ADP and inorganic phosphate. Okay? Now, this is how the process occurs. The process occurs on the thylakoids. We have the outside of the thylakoid and the interior of the thylakoid, which we call the thylakoid lumen. Now, when photolysis occurs, these protons accumulate in the lumen of the thylakoid. So we shall have a number of protons inside the thylakoid. Now, when these protons accumulate, they create an electrochemical gradient because we have more protons inside and less on the outside of the thylakoids. So what happens? There is an electrochemical gradient that is created because this membrane of the thylakoid is impermeable to the hydrogen ions. So for them to diffuse out, they pass through the special proteins that are associated with ATP synthase enzyme. So as the protons move or diffuse from the interior of the thylakoids to the outside of the thylakoids, they disturb the structure of these stalked particles that are associated with ATP synthase enzyme. Therefore, this one provides energy that is required to combine ADP and inorganic phosphate to form ATP. This is what we call photophosphorylation, the addition of an inorganic phosphate to ADP to form ATP by using energy from light, and in this case the sunlight is called photophosphorylation. Okay, so this ATP produced is made available to the light independent stage of photosynthesis, as we shall discuss in our next lesson. Okay, now these protons here do not end here, but they are also used to reduce NADP, as we are yet to see. Here, when a photon of light strikes photosystem 1, the electrons within the centers of photosystem 1 are also excited and they are boosted to even a higher energy level, where they are accepted by another electron acceptor. Now, These electrons may be returned back to photosystem 1 along the electron transport chain, where they can also be used in the generation of more ATP, and that is called cyclic photophosphorylation, because electrons will have moved in a cycle. You can see from photosystem 1 to electron acceptor, then along the electron transport chain back to photosystem 1. However, the electrons here may also be handed over to other electron transport chain here, and in this case, these electrons together with these protons are used to reduce NADP in the presence of an enzyme NADP reductase, and this one results into formation of NADPH2. This NADPH2 is a reducing agent that is used in the Calvin cycle, or the light independent stage of photosynthesis. Okay, so in our next lesson we shall discuss the light independent stage of photosynthesis and the role of NADPH2 and the ATP produced in this cycle. Don't miss it. Nice time.